Hey, good evening, students. This one is the chapter number 9, which is called as the biotechnology. Actually, it is a chapter number 8 right now because first chapter is missing now. So, biotechnology it divided into two parts. This chapter is the first chapter, and the second chapter is chapter 9, that is chapter 10. So, biotechnology, principle and processes is the first chapter. It is divided into total parts, 8 total 8 it is divided. So not going to spending so much of time, we have to start this chapter. In this chapter, the first part is what is biotechnology. Actually, it is a type of a study where a living organism which produce a product, and the product is going to be produced by any living organism or formed by a living organism for the helpful for human beings. The product may be of any type, like as a test tube baby, like as a gene product, like as a vaccine, or any type of the defective gene correction. This is the first part. So what the question is arises, what are the product of these, what are the product formed by the living organism? Why biotechnology is important? Many times the question arises. The second part is the principle. There are two type of the principle on which the biotechnology is based. Number one is genetic engineering, and number two is bioprocess engineering. If it is a genetic engineering where the DNA and RNA are inserted inside the host to change the phenotype of any type of the host, and the bioprocess engineering is the product which are obtained by the help of the microbes which are grown in any condition, and the products may be antibiotics, vaccine or enzyme. Now the third part. The third part is actually recombinant DNA technology. The recombinant DNA technology, here I tell you the recombinant DNA technology in a six part, in a six step. The first one is the restriction enzyme cut the desired gene or DNA. Then these genes are arranged in size by the help of cell electrophoresis. This gel electrophoresis consists of a size where the restriction enzymes, the third part is the restriction enzymes cut the plasmid. Plasmid is actually a double standard circular DNA which is present inside any bacteria or any host from where we obtain this plasmid. So restriction enzyme cut the plasmid. The gene which is present in the electrophoresis, that is desired gene, joined with the plasmid by the help of ligase, and then this become a recombinant DNA which is inserted inside the bacteria, and bacterial cell vector like bacteria cell act as a vector like as a cosmid, phasmid, artificial chromosome, and all of them. So this is the third part. Now what is the fourth part? The fourth part is we know about the plasmid, but what is actually a plasmid? A plasmid is very small, circular in piece. It is found in bacteria, has at least one gene which is going to be the desired. Can fix nitrogen. Larger plasmid consist of low copy number. They can replication and of five types: resistance plasmid, call plasmid, valence plasmid, degradative plasmid, and fertile F plasmid. So the next part is fourth one. What are the tools used in the recombinant DNA technology? The tools which are used in recombinant technology is four. Number one, restriction enzyme. Number two, ligase. Number third, vector, and number four, host. The question maybe arises, what are the tools of recombinant DNA technology? The first one is restriction enzyme. The first restriction enzyme name is Hind2, which cut any type of the DNA in the 6 pair of a gene. Nowadays, 900 restriction enzyme is obtained from the 230 strains of a bacteria. And each restriction enzyme has consist of first letter which is comes from the genus of bacteria from where it is obtained, And second and third is the prokaryotic cell from where it is isolated. Like as a E. coli. So ECO R1 coming from the name of E. coli bacteria. So there are two type of restriction enzyme. One is exonuclease and another is endonuclease. Exonuclease cut the DNA from the end part only, and endonuclease cut any specific sequence which is palindromic. Palindromic means like as a dalda. In from right hand side, from the left hand side, the same perfect letter is going to be formed. The next part after restriction enzyme is ligase. Ligase is the glue joining the Okazaki fragments. It repairs the DNA broken parts as of four types. One is DNA ligase 1, second DNA ligase 2, third is DNA ligase 3, and fourth is DNA ligase 4. Now the next part is a cloning vector. Cloning vector clone the DNA and make the copies. Some form 2 or 3 copies and some form the 50 to 100 copies. कुछ 2 से 3 copies बनाते हैं, कुछ 50 to 100 copies. Everything is perfectly right here. They have a 4 specific part. One is ORI region, another is second one is restriction site, third one is selectable marker, and fourth one is transgene. Now what is ORI region? ORI is the origin of replication which allow the vector to replicate inside the host. Restriction site is the part where the foreign DNA can be inserted. Matlab, it can be cut by the help of the restriction enzyme, and the new DNA, which is that is the interested gene, which coming from outside is going to be inserted inside it. The selectable marker is a gene which help to identify if the cell is going to transform with correct plasmid or not. Like as example, ampicillin, chloramphenicol, tetracycline. The restriction site name is also like BAM-H1, HIND1, SAL1, like this. And the transgene is the DNA which is going to be inserted inside. So fourth thing is going to be there. The seventh part is actually the host. अभी ये सारा काम recombinant DNA is formed. Now host, the cell, the bacterial cell which actually act as a host. DNA is a hydrophilic molecule जो कि pass नहीं होता है through the cell. So to treat by the help of the divalent cation like calcium, DNA can be passed inside the cell by the help of three methods. One method is called as a heat shock, where the DNA firstly cooled down in a low temperature and then inserted inside the cell and heat it at 45 degrees Celsius suddenly, and then suddenly cool down by the help of the ice. So this up down temperature create a distraction and problem inside the DNA, and because of that the heat shock is going to be happen. If this type of the process is not there, then microinjection is there. In the microinjection, the recombinant DNA is inserted by the help of a microinjection. This is called as a microinjection method. And the last one is ballistic gene gun, where the cell which is going to be inserted is going to bombarded with the microparticles, and these microparticles are coated with the DNA. Suddenly when you hit in the high speed, it goes inside the cell and attach there. The armed DNA used to disarm vector. Overall, last part but not the least, actually it's second last part, how the recombinant DNA technology is going to be used, and don't miss any step. Firstly, the DNA is taken from any part and isolated by any enzyme like as a ribonuclease or protease. DNA is cut by the help of the restriction enzyme, restriction endonuclease. The cutted part transferred to the agarose gel, where the gel electrophoresis technique is going to be happen. Here the DNA is moved towards the positive terminal because DNA is a negative part. So short fragments moves faster to the positive part and larger fragments slowly move. All the part is now cannot be seen by the naked eye, so it can be stained by the help of ethidium bromide, which is a type of a staining part, and this staining ethidium bromide can able to show the DNA under the ultraviolet light. This ultraviolet radiations clearly show the DNA fragments, and we take the fragment from there and separate the bands and cut out the agarose by the help of the process called as elution. Then this part is going for the amplification. Amplification is a type of process in which any DNA of interest is going to take place and make the copies of it. So there are two parts, two process. One is DNA polymerase enzyme is used, and second is DNTPs. So double standard DNA, firstly denatured at 95 degrees Celsius for 15 seconds. Because of that, the both strand of the DNA is going to be separated. A set of primer is going to attach to the template strand used to anneal the single strand. Because of the help of the Taq polymerase, new strand is going to be formed in the extension process. This extension process forms the DNA and transfer it to the vector. But before that, ligase come to join the defragment or disjoint part of the DNA. Now, newly formed R recombinant DNA transferred to the host cell, and this host cell is going to cultured in the foreign gene product. And lastly, the copies is going in the worst way. So lastly, downstream processes is going to be taken place by the help of a bioreactor. Bioreactor is of two type. One is a simple stirred tank. Another is sparse stirred tank. Sparse में फेना means just like as example fog होती है उसमें, and in simple it is going to be distilled like that. The digester is also there, purificator is also there, other. So downstream processing is, firstly the DNA going to be taken out, is separated, and then it is going to be purified and then published in the market. So this is the chapter of principle and process. After that, the gene product. As you see, bioprocess engineering is coming. Don't worry, go through the chapter, and the question will be coming very soon. PYQ is not going to be attached with that, because I am going to show you the question and direct the question after 25th. And after 25th, be stick with your mobiles. Thank you. Have a nice day. Like, share, and subscribe. And definitely read and understood the chapter very clearly. Thank you. Have a nice day.